Thank you very much. And first, I'd like to thank the organizer, in particular Kotori-san, for giving me an opportunity to be here. Well, as she briefly explained, I'm coming from Kyoto, and actually I joined one of the WPI program in Kyoto University, which is ASHBi, and that is studying the human biology. And actually, I'm working on fusion researches there between mathematics and biology. And I'm one of the mathematics PI, and also I'm a vice director there, mainly in charge of the fusion research. So today I'm very happy to share some of the experiences so far I got, in particular about the fusion researches in the WPI program. And my title is Some Fusion Research Between Mathematics and Life and Material Science. So this is the basic my plan of my talk today. So I roughly divide into two parts. Actually, so now I'm in Kyoto, but before Kyoto, I was in Tohoku University. Also joined to another WPI project there. So it is called WPI-AIMR, this one. And this institute mainly studies the material science. And I was working in that institute too to promote the fusion research between mathematics and material science there. And then after Tohoku, I moved to Kyoto about 6 years ago, and we launched new WPI project in Kyoto that is called ASHBi, studying the human biology. So now I'm interested in collaborating with biologists about fusion researches with mathematics and biology there. And well, I belonged to the two WPI, but my main interest for this period has been developing the novel data analysis method by using mathematics for material science and also biology. As you know, now we can get a bunch of massive and complex data, even in both material science and life science. And my interest is to develop new data analysis methods for those massive data by using advanced mathematics. So in Tohoku University, I was interested in the material structure analysis, and then there I mainly used topology to understand structures and also relationship to the functions of the materials by using topology. And then in Kyoto, I'm interested in single-cell biology. And we are now developing several tools, mathematical data analysis methods based on probability theory and optimal transport theory, as well as topological data analysis too. So today, Kotori-san asked me to give a talk here, and she suggested that in view of the diversity of the member here, WPI in Hiroshima, it may be nice to not only focusing on one topic, but also to talk about both topics and share my, well, limited, but my experiences about fusion researches in the WPI program. So I will talk about two topics, but although each topic may be short, but I'm very happy to discuss more in detail if you are interested in some of the topics after, say, my talk or Q&A session. Okay, so let's move on to the first topic. So this is about the material science and the mathematics fusion research. And the main topic, well, not topic, sorry, main tools, in a sense, tools from mathematics in that fusion research is the so-called topological data analysis, TDA for short. So, well, TDA is a relatively new research area in mathematics. Actually, it was developed or we got a foundation in this century, actually. And we have a kind of slogan saying that data has shape, and then shape has meaning, and the meaning drives values. So this is a kind of slogan in TDA. And under this slogan, we are developing many tools for analyzing massive and uncomplicated data to characterize the shape of data. And I think the idea to apply topology into data science or data analysis is, at least for me, it looks very interesting in the following sense. So suppose you have a big data, complicated, massive data. As you know, topology is one of the branches of geometry which allows us to think your object coarsely. It allows us to consider continuous deformations. So we can get very simplified structures with keeping topological information. And the idea of TDA is to apply such idea to the data analysis in the sense that suppose you have a big data, by using topological tools, we can get the essential structures by viewing your object in a coarse-grained way. Based on such a coarse-grained way, we can get essential data structures from the big data. So this is a very rough idea how to use or how to apply topology in our data in the big data or massive data. And based on such a concept, now TDA is studied very actively from both theoretical way and also application ways. And I'm here listing some of those applications. So first, material science. I think this was initiated by our group in AIMR. And also it is applied to neuroscience and genome science, and also protein structural analysis and information theory, and more generally, big data analysis, including social network analysis, healthcare, developing the medicines, stock market, image analysis, etc., etc. Yeah, so this is a rough sketch of the background of the TDA. And actually, I want to mention here that protein structure analysis, as far as I know, the first application of topology into the protein structure analysis was done in here, Hiroshima University. Actually, at that time, I was an assistant professor here. And there was one professor, his name is Izumi-san, Shunsuke Izumi. He's still in the Faculty of Science member. And he was so friendly and he was so aggressive to apply any types of mathematics into protein structural analysis. And I had the opportunity to chat with him, and we started protein structural analysis using topology. So in that sense, I'm very happy again to be here to explain or to give my talk here. Anyway, so this is a brief overview. And I'd like to move on more details about what we have done in Tohoku University. So this slide shows our summary of the results done in Tohoku, mainly supported by AIMR and several other grants, including national project in material science. And also we did a lot of collaborations with companies based on TDA. So we mainly worked on glass structure analysis. And also we worked on protein structure or polymer structure analysis. And we collaborated on the, say, battery materials or rubber materials and so on. And in the middle, we also added tools from machine learning to TDA by which we can predict the relationship between structure and the functions of the materials. And then today, I'm gonna talk about some of those topics in the first half. Okay, so the main tool, the main tool used in materials TDA project is the so-called persistent homology and persistence diagram. And then here I'd like to explain these two concepts in intuitive way and also mathematical way separately. So first, this is a slide for intuitive explanation about persistent homology and the persistence diagram. So first, very intuitively speaking, persistent homology is just a machinery to assign each input data. So given input data, so suppose your input data is, for instance, atomic configurations of materials. It is just a point cloud, just a point in the space, or digital images captured by, say, microscopy and so on, digital images, or time series data, so whatever. Given input data, persistent homology outputs the so-called persistence diagram, PD, to each input data. And the PD consists of some points in the half plane above diagonal. And this diagram, PD, contains the information of shape of data, shape of input data. More precisely, it characterizes holes in our data, like topology likes to detect holes, describes number, size, and the shape of those holes in a multi-scale way. This is very important. We don't need to restrict our attention in single resolution or single scale, but we can do our analysis in a multi-scale way. So this is the input and the output correspondence of the persistent homology. And as a user, it is enough, or it suffices to understand the meaning of the persistence diagram. Once you understand the meaning of persistence diagram, you can skip all the details, all the mathematical details hidden here. You can use it. Actually, most of the collaborations with, say, companies and material scientists, they don't understand. They don't need to understand inside of this mathematics part, but they try to understand the meaning of the persistence diagram. And once they understand, they can use this tool. And let me explain the meaning of the persistence diagram here. So suppose, just for simplicity, suppose you have these 4 points as your input data, just for simplicity. Suppose you have 4 points in your input data. Then first you put a ball on each point like this, and increase the radius of those balls from very small to very large. You consider a fattening sequence of balls. That is called filtration in mathematics. Then at this specific radius parameter, say B, hole appears here. And we record this specific radius parameter B as bars of hole. And then you further increase radius. Then this specific hole, this hole disappears or covered by this radius parameter D. And we again record this radius parameter as this. And for each hole, we plot this birth-death pair in a plane like this. Birth axis and then death axis. And then persistence diagram is just a collection of those birth-death pairs computed for all possible holes in your data. So just superpose all the birth-death pair in the half plane above diagonal, like this. So this is a persistence diagram. So from this construction, each point in the persistence diagram corresponds to a hole in your data. And the birth and death axis measures shape of holes. And importantly, points close to diagonal, like these generators, we can mathematically prove that those holes can disappear when you add small perturbations to your input data. So in a sense, it is a noisy generators, noisy holes. On the other hand, point away from diagonal like this, we can mathematically prove that this hole is stable with respect to small perturbations. So not only counting holes, but also we can get a metric, in a sense, metric type information. You can get metric type information from the persistence diagram. And so this is showing the example given atomic configuration of hemoglobin. And then this is a persistence diagram of that. Then, as we see, there are a bunch of small generators close to diagonal. And from this exposition, those are very noisy ones, like small holes there. On the other hand, you can see this point here. It is supposed to be, it should be stable with respect to perturbation in the hemoglobin. And this actually corresponds to this hole. So this is just a demonstration, but given your massive data, we can characterize our data in this way. So this is the essence of the persistence diagram, and then persistent homology, and then persistence diagram. So far, so good. Okay, so this is intuitive explanation of the persistent homology and the persistence diagram. Yes.
So what is the prediction of the bus radius? For example, in the figure, you take the average.
Take what?
Minimum in the bus point here. So because there's some overlap between
So the definition of the burst radius is the radius at which we can get this hole. Suppose that this radius is slightly shorter than that, then we cannot get a hole, precisely speaking. But at this specific value, we can get. Yeah. So please interrupt me if you have some questions.
I have one question. If now you draw the circle, but in the data space, it's not necessarily circle with some atomicity.
Yeah.
How does it affect the diagram?
Well, the short answer is that the output will be different for each choice of your shape. First of all, mathematically speaking, this component, disk or elliptis and so on, those should be convex shape in order to keep topological information. But once you have a convex object to get a filtration, at least you can get a persistence diagram which captures the real topology. But the output of the persistence diagram will be different depending on your choice. So it's up to your selection, in particular, depending on your question. For instance, this is somehow the most simple way to get a filtration. But if you consider this ball as I don't know how to say in English, electron cloud around some nucleus, then it may be natural to consider ellipsoid point maybe. But this is one side to answer your question. But on the other side, for the purpose of computation, it will be very difficult to get a persistence diagram using elliptis point. Yeah. So yeah, it's a kind of trade-off which you use. Or this is explained basically using the point cloud data. But suppose you have a digital image data. In such setting, the basic structure is a cube or pixel. In such a setting, we replace ball with a pixel. But then, even in the sense of the computations, we can get persistence diagram very fastly. Computationally, it is very easy to get. So perfect circle or pixels are, I think, one of the most practical solutions, I think, to get persistence diagram. Okay, so I don't know how I need to explain persistent homology and the persistence diagram in mathematical way. Just briefly, I will explain. So this slide shows a mathematical definition of the persistent homology and the persistence diagram. So the input will be the filtration of the topological space, like a patterning sequence of the simple shell complex or cell complex and so on. Then persistent homology is just algebraically speaking, it is just a sequence of homology vector space. Homology vector space on each xi connected by linear map induced from inclusion. So just it. So this is very simple object in mathematics. And such an object has a nice property. It is called the interval decomposition property. It is actually coming from Gabriel's theorem. It says that any persistent homology under a certain mild condition, any persistent homology can be uniquely decomposed into the direct sum of the so-called interval representation I. And here I BD means that we have a one-dimensional vector space from XB to XD connected by identity map and a zero otherwise, like interval starting from B to D. And topologically speaking, of course, each interval I from B to D means that at XB, topology hole appears, and it persists up to XD in the filtration and disappears. So each interval expresses such an event in this process. And then, given such a unique decomposition, persistence diagram is just an invariant, algebraic invariant, in the sense that we just gather all the birthday pair appearing in this decomposition. And then just plot it, then we can get persistence diagram. So this is a rough sketch of the persistent homology and the persistence diagram in a mathematical way. Okay, so now let me apply it into the material science. So actually, in Tohoku, we have applied TDA in actually many materials. But this was the first result obtained there. And the tools or methods developed in this first work was mostly still even now used in the materials TDA project. So in this research, we are interested in understanding the hierarchical structures in the glass materials. And well, in particular, we are interested in the geometric structures only embedded in the glass states. So here, I'm showing 3 atomic configurations of the material silica, SiO2, meaning that each point corresponds to silicon atom or oxygen atom in a crystal state, glass state, and a liquid state. And well, as you see, it's easy to distinguish crystal state because of its periodicity, right? It's easy to distinguish. But it's not so easy to distinguish between glass and a liquid state by just looking at them. Both look just random. But as you know, glass is a rigid material. It means that it has to have some rigidity. It has to have geometric constraints to achieve, to realize such rigidity. And this is what we want to understand. So we want to understand some hidden structures there to realize such rigidity. This was our starting point. And we applied the persistence diagram, and these are the persistence diagram of them, meaning that we just consider atomic configurations as point cloud, just a point in a space, and we put a ball on each atom, increase the radius parameters, and measure the birth and death, as I explained. And these are the results of persistence diagrams. And we see that in a crystal structure, persistence diagram consists of some of the isolated islands, like zero-dimensional support. In a liquid state, persistence diagram consists of points scattered in a two-dimensional space above the diagonal. But in a glass state, we can see curve, right? We can see very characteristic curves. And it says that at least persistence diagram is sensitive enough to detect glass states. And next, our interest was to understand the hidden geometric structures there, only embedded in a glass state. It means that we are interested in the original geometric structure which generate those curves. This is what we want to understand. And this is nothing but the inverse analysis, right? So now we are asking, given this persistence diagram, our interest is to find the specific structures there which generate those curves. And this is just an inverse analysis. So we have developed a mathematical method to solve the inverse analysis or inverse problem on persistence diagram. This is one of the fusion research done in Tohoku University, motivated from the discussions with material scientists. They really...
I have a question there. So in gas state and liquid state, the particles moving around and dependent. How How did you calculate your resistance?
Basically, first of all, I need to mention that those atomic configurations are obtained by molecular dynamics simulations, and then physicists and material scientists prepared atomic configurations in those states which are almost in the stationary state. In that sense, we have time fluctuations of the positions of the atoms, but then those are supposed to be just small heat fluctuations, not drastic changes. So in that sense, persistence diagram there is computed in the average sense. We prepare a time scale, and in that time scale, positions are slightly changing by the heat fluctuations. But on each time, on each time in that time scale, we compute persistence diagram, and this is a result of the averaging of them.
Kind of an ensemble.
Yes. So we developed an inverse analysis tool, and we found that actually those curves are really important to understand hierarchical structures of the glass. More precisely speaking, the rings sitting here are the primary rings, which generates secondary those curves. And the positions of the rings are in the sense of the hierarchical locations. So this will be the primary ring, and inside of this primary ring, we can get secondary rings, and those secondary rings are corresponding to those curves coming from the diagonal. So we found this kind of structures, and also we further worked on the understanding the relationship between the existence of these curves and rigidity. Very intuitively speaking, existence of curve is saying that this point can move along the curve by the heat fluctuation, but this ring cannot move to the normal directions. So the existence of curve is actually giving us one constraint, codimension one constraint, and which is expected to be somehow related to rigidity of the materials. This is an intuitive idea, and we actually refined this statement by using molecular dynamic simulations and this inverse analysis technique.
So in your diagram, so the material is a kind of a ring arrangement. So that kind of donut shape. So the donut shape is always keeping in the dynamics of the glass state.
You mean?
So
this shape is preserved in the heat fluctuation?
Persistently existing in the glass state, keeping just a donut shape or not.
But so this is what we want to understand. Actually, shape itself is slightly changing, but there's some specific directions of the deformation which are not allowed to be deformed. And this direction, prohibited direction is corresponding to the normal directions of the curves. Normality, it means that we cannot move to that direction. So such a prohibition is actually translated in the shape level, and we actually checked such a correspondence by careful studies.
So those particles tended to keep this line.
Yes. Yeah.
See, I got it.
So I honestly, I need to explain more to explain more details. But if you are interested, please look at this paper, or I'm very happy to share more details after my talk. And so we developed software. Software development is also important to accelerate fusion researches. And once we have developed software, many material scientists, not only in AIMR, but also companies guys or other universities, they started collaboration with us. So this is free software, so you can download it. And we also recently wrote a paper explaining how to use this software in the material science in this journal. And also, at the end of my first half of my talk, today I only explain glass materials, but once we developed mathematical tools, because of the nature of the mathematics, we can apply that tool to other materials too. Yeah, horizontal expansion is a good advantage of the mathematics, I think. So we can apply it into battery materials, magnetic materials, rubber materials, those CFRP and protein and so on, including companies. So this is what we have done in AIMR. And actually, I would like to emphasize that during AIMR, Kotani sensei, Motoko Kotani, she was at that time she was a director of AIMR, and all of these collaborations with material scientists. Well, of course, it took a long time to get the results, but Kotani sensei kindly supported our activity, and I thought that it is really, really important to have a continuous support from the director. You know, saying fusion research is really very easy, but doing fusion research is not so easy, actually. And yeah, continuous support from the director was, at least for me, so important to focus on the fusion research in AIMR. So I really appreciate Kotani sensei again here. Okay, so this is the end of my first part of my talk. And I'd like to switch to another slide. Okay, so in the latter half of my talk, I would like to explain my recent activities in WPI in Kyoto, which is called ASHBi, Institute for the Advanced Study of Human Biology. So now from materials, I switched to biology. And the theme and key research goals of ASHBi is summarized in this slide. So first, the main question in ASHBi, Human Biology Institute, ASHBi is this: what key biological traits make us human? And how can knowing these lead us to better cures for disease? So this is the main question raised by ASHBi. Well, this is a very big question to answer. In particular, this one, what is human? This is a very big question, but to that big question, ASHBi investigates a key concept of human biology with a particular focus on genome regulation and then disease modeling. And these 5 are the key research goals. Well, there are several goals, but mathematics team basically focuses on number 2. So we would like to clarify the principles defining the species differences and the human traits. So this is a question assigned to us by mathematicians from the director. And so this is a global picture of ASHBi organizations. So now we have, I think, 17. Yes, we have 17 PIs, mostly coming from biology side. But we have one ethics PI, Fujita-san, and we have 2 mathematics PI. One is me, and the other is Seirin-san. So this is a global pictures of ASHBi. And in the mathematics group, so basically our group, left-hand side, is working on like AIMR, like the work in AIMR, we are developing novel data analysis tools using advanced mathematics for biology. And the other group is basically doing the mathematical modeling. And we are actually seriously collaborating with biologists listed in the previous slide. And so this is a kind of flagship project. Set it in our mathematics group. So we would like to establish the so-called data representation theory. And to that goal, we established a new mathematics so-called data representation theory through fusion research with biology. So first, you'd like to develop mathematical descriptors, mathematical data descriptors like persistence diagram that characterize essential structures in massive and complicated biology data by using geometry, probability, and then as well as topology, as we did in material science. And then we'd like to understand mechanisms behind phenomena that generate those data, biology data, by mathematical modeling. So this is what we are now working on. And then today so since I already talked about TDA, topology, in the rest of my talk, I'd like to focus on the first probabilistic data analysis method, and then geometric data analysis method. So first, probability part. So let me first briefly explain the single cell data, the properties of the single cell data, and some of the problems known as curse of dimensionality. So I don't know how much you are familiar with single cell RNA sequencing data and single cell data, etc. But very briefly speaking, now single cell data is actually very getting popular in current biology. Actually, that is, in a sense, revolutionizing the concept of data. So we can get massive, very massive data. For instance, scRNA-seq data provides us with this kind of matrix data. It consists of gene by cell matrix for a given cell population, gene and the cell matrix, meaning that each cell viewing column way, on each cell, we can measure the expression value of gene expression values on each individual cells. So this is one of the types of the single cell data called the scRNA-seq data. And it has some properties. The first one I should mention is that it is very high dimensional data, meaning that usually the number of genes is treated as a dimension of the data. And since our interest is human, our institute is a human biology institute, our interest is human or more generally primate, then the number of genes is something around 20,000. So the data itself is very high dimensional data. And of course, it contains technical noise in the process of sequencing caused by experiments. Then in mathematics, such a data means high dimensional data with noise, is frequently suffering from the so-called curse of dimensionality, which causes adverse effect, bad effect in the downstream data analysis. In particular, we cannot measure the correct distance between 2 points. So this is a very serious problem, right? Because most of the data analysis methods, say clustering, for instance, clustering is actually using the distance information. But the curse of dimensionality breaks such a distance information very easily. So let me explain how COD is giving us a serious effect in our data, in a downstream data analysis by using example. So suppose you have 8-point clouds located at the vertices in the 3-dimensional rectangle like this. Then we embed this data into D-dimensional space with tiny noise. And we increase D from very small to very large. And let's see how the downstream data analysis is affected by COD. For instance, let's use t-SNE. t-SNE is one of the most famous methods for clustering. Then when d equals to 3, embedding dimension is 3, we can perfectly recover 8 clustering as it is. This is good. But when d equals to 1000, a kind of averaging behavior occurs. It means that now pink and purple cannot be distinguished, right? Red and orange are merged together, blue and green, gray and brown. So now we can only see 4 clusters. This kind of averaging behavior occurs, and this is nothing but the effect of the curse of dimensionality, since we cannot measure the correct distance. And when d equals to 20,000, which is almost the same as the number of genes in human and primate, we can only distinguish left and right. So this is what we are observing, actually, in the downstream data analysis. You may here think that, all right, COD is caused by the effect of the high dimensionality. So what if we first apply PCA into low dimensional space and do the downstream analysis? That may be a good resolution of this problem, but short answer is not. That is not sufficient. Actually, PCA itself suffers from COD. This, and as these 2 panels shows. For instance, this blue panel shows that using PCA in 20,000 dimension, we completely lost the data structure. This to this. So this is nothing but the effect of the COD. So this is really a big problem. So CODs mask through single cell structures. And this is, in a sense, ironic situations in the following sense. Technologically speaking, we are getting single cell information. We have a tool, experimental tool, that provides us a single cell data. But because of the lack of mathematical method, we cannot observe single cell structures hidden in your massive data. Right? So in that sense, mathematician has to work more to follow up the recent progress of the technology. And this is what we want to solve in ASHBi. So we want to develop a method which has real true single cell resolutions. And then we developed a method so-called record. This is a collaboration with our director, Saito-san, with our postdoc, Imoto-san. He's a mathematician. In essence, given single-cell RNA sequencing data, our method, record, reduces technical noise in SER-seq data with UMI counts, and it actually resolves CODs by using high-dimensional statistics theory and probability theory. So this is a first, this is a summary of the result, and there are so many mathematics behind this, but I cannot explain them here because of the time restriction. But so let me explain how we conducted fusion researches with biologists about this project. First, we thought that this problem is really important, as I explained in the previous slide. So first, we really wanted to understand the noise generation mechanism, the mechanism, how noises are added in your data. So we really wanted to understand that process. So we had many discussions with biologists in ASHBi. And after those discussions, we found that the essence of the noise generation can be described by the so-called random sampling process in a probability theory. Given your sample, and then you get a randomly, then at certain chance, you may lose to pick up. And that is almost the same process what is done in the SER-seq data sequencing process. So by using such an understanding, we could get a really good estimate of the momentums, like an average and a variance and so on of the noise distributions. And once you get the momentum information of the noise distributions, we can apply the high dimensional statistics theory to remove. And we succeeded in removing the curse of dimensionalities. So this is what we have done. And well, our method doesn't have any parameters. I think this is good for practitioners. We don't need to tune parameters as you want. No parameters like PCA, and we don't need any averaging process. Usually conventional methods use averaging, but recall that our interest is to get a data analysis method which has single cell resolutions. Averaging breaks such a resolution, right? We want to get a real single cell resolution, and our method doesn't include any averaging. Well,
in terms of the noise distributions, I'm afraid that so in a single cell analysis, that each cell should have some different noise distributions. Even the instrumental part is exactly the same approach is applied, but
yeah, basically we have 3 parts in the experimental process where we get noise, which are supposed to be independent of the selection of cells. The first one is a sequence. I hope I can explain. The first part is a sequencing part, and the second one is an amplifying part, and the third one is I forgot, sorry. But there are basically 3 parts where noises are generated, and we thought that at least moment of the error generations are almost independent of the selection of the types of the cells.
Really. So if we apply to the different types of the cells, the noise generations are almost the same.
Yes. Basically, we are thinking about the amount of the messenger RNA.
Right.
And that are, at least in our mathematical modeling, we don't care about the types of cells.
Oh, really?
If such a dependence is actually important, we may need to tune or modify our mathematical modeling. But at this moment, we don't care about the difference of cells.
So once you determine the noise distribution in some particular cells or cell lines, so you don't need to tune to the other cell applications.
Let me explain. Maybe there is some misunderstanding. Suppose you have cell population which may include different cells, not only one single cell, not only one type of cell, but several types of cell. But suppose you have one cell population, we can determine the moment of the error distribution. This is what I want to say.
I see.
Of course, if you change the cell populations, if you change the lines, experimental lines, so on, noise moment maybe change. Well, I think will be changed. But once you have cell population, then we can at least measure those moments.
In the same populations, only one noise distribution.
Yes, that is what I want to say.
Otherwise, very difficult to apply. Every time we have to check.
Yeah.
Okay, I got it.
So let me move back to biology. So this slide shows how record is. I have 10 minutes, still 10 minutes, including, I think, Q&A. So this slide shows how record is working well to remove the noise and then to get a single cell resolution. So let's see yellow panel. This yellow panel, in this yellow panel, we prepared 5 cell populations using primordial germ cell-like cell. So it's a kind of germ cell, and also somatic cells. And we prepared 5 groups. The group number 1 consists of 1,000 germ cell PGCLC plus 3,000 somatic cells. Group number 2 consists of 30 PGCLC plus the same number of somatic cells, and so on. And group number 5 consists only 3 cells, 3 PGCLC plus the same number of somatic cells. So this is we prepared. And we measured the expression value of gene SOC17, which is supposed to be high in germ cell, but supposed to be low in somatic cells. And we applied our method record and also two conventional standard methods for noise reduction in single cell data. We see that for magic and enhanced conventional methods, when the number of germ cell decrease from 1,000 to 3, the expression value of SOC17 is also decreasing, although it is supposed to be high in PGCLC. And this is nothing but averaging effect, because suppose in group number 5, you have only 3 germ cells in 3,000 somatic cells. And as I said, SOC17 is low in somatic cells. Now, 3 cells are just a minority. Because of the averaging effect, SOC17 value is very low. It means that by using magic and enhance, we cannot detect germ cell losing single cell resolution. We cannot detect 3 cells. On the other hand, record, even 3 cells, the expression value of SOC17 persists, meaning that we can detect 3 cells by using record. I think it actually shows the single cell resolution in our data analysis. And also, now since we have a single cell resolution, we can see more detailed structures of the biological process or cell differentiation process. Let's see this figure. We applied our method to some public data, mouse data, and applied t-SNE. Before record, we can only see 3 connected components, just 3 islands, very rough structures. But after record, we can see more finer structures. In particular, we can detect the existence of this kind of pathway passing through the so-called notocode. This is actually a new biological finding. Yeah, so this is the essence of the record, and for details, we have applied this method to several biological problems. So for detail, please look at this paper. And actually, from the start, it took almost 5 years to get this result. From the viewpoint of evaluation, it may not be a good strategy to take 5 years to get one paper. Actually, I took 5 years. But the director, Saito-san, he understood the importance of this method. And he seriously thought about the importance of the fusion researches. And like an AIMR, again, I got a really good trust, a kind of trust from the director, and we could get a really good relationship. And that was very important, I think, to focus on fusion research. Yeah, so this is one fusion research done in ASHBi. So I really appreciate Director Saito-san. And in the rest of my talk, I have 5 minutes. I would like to quickly review the geometric data analysis method using optimal transpose theory. Again, problem is coming from, well, our tool is totally motivated from the biological request. You may know that very famous biologist, Waddington, proposed the so-called landscape pictures to explain the cell differentiations, like this figure shows. So in this model, or in his model, this is a very conceptual model, but his idea is that cell differentiation is proceeding like this figure, meaning that stem cell is located at the top of the hill. And when the cell differentiation proceeds, this ball is rolling down the hills and then converges into some valleys like this. And each valley corresponds to the cell type, say kidney cell, neuron cells, or germ cell, and so on. This was his picture. But the question is that what is the actual shape of Waddington's landscape in real cell differentiation? This is just a conceptual model, but we want to describe actual shape to understand more details about dynamics. And what are the marker genes that shape those values? So this is a question raised by biologists in ASHBi. And to that question, now we can use time series single cell data, which are supposed to include information about dynamics of cell differentiation. So by using such a data, we want to develop a comprehensive data analysis method to study dynamics of cell differentiation. So this is what we want to do. But the problem here is that there's a serious problem using snapshots or time series data of single cell data. The problem is that at each measurement, at each time, cells are killed. It means that even if you have two single cell RNA sequencing data in adjacent times, we don't know how to connect these cells together. We don't know this cell in time 1 is corresponding to the counterpart in time 2. We don't know this kind of natural matching. And this is the problem, but the current mathematics, one of the current mathematics, so-called optimal transport theory, provides a really good tool to, I don't say answer, but to attack this question. And we applied optimal transport theory to develop a data analysis method for the time series data. And this is the summary slide. This is the last slide. So we developed a method so-called SC single cell EGOT based on the Gaussian mixture optimal transport theory. So this is a pipeline, what SC EGOT is doing. Your input data is, as I explained, time series SC and SEC data. Then SC EGOT first describes your data by the Gaussian mixture model, summation of the Gaussian distributions. And then we developed the mathematical theory of the optimal transport under the setting of the Gaussian mixture models. And then such a mathematical foundation, we can get a time continuous interpolation between each Gaussian distribution, from which we can get a correspondence between cells in adjacent times. So this is a rough picture. And actually, SC EGOT outputs will provide us with 6 outputs: cell state graph that can be used as clustering to understand cell differentiation. We can also get velocity. You may know that RNA velocity is a very famous method in biology to study dynamics. We can get a velocity like RNA velocity, but the advantage of our method comparing to RNA velocity is that in RNA velocity, they need additional information of the splicing and the unsplicing data, meaning that that method can be only applied in the RNA sequencing data. But our method doesn't need that. It means that our method can apply not only RNA sequencing data, but also SC ATAC data, SC high-C data, epigenomic data, and so on. So this is one of the advantages of our method. And since we get a kind of vector field in the terminology of mathematics, we can reconstruct dynamical systems based on this vector field, from which we can construct gene regulatory network. And also, if your biological system can be supposed to be the gradient system, we can reconstruct a potential function by solving the some kind of partial differential equation, and then we can get a potential. Actually, this is supposed to be a Waddington's picture I explained in the previous slide. Yeah, so we recently uploaded our result in this bioarchive. So if you are interested, please look at that. Okay, thank you very much for your kind attention. I will stop here.